Resuming page 92. DNA replication. DNA replication is the process by which two new strands of DNA are made, using the two existing strands as templates. During interphase, DNA and its associated proteins appear as dispersed chromatin threads within the nucleus. When DNA replication begins, the two strands of DNA molecule separate from each other for some distance. Each strand then functions as a template or pattern for the production of a new complementary strand of DNA, which is formed as complementary nucleotide pairs with the existing nucleotides of each strand of the separated DNA molecule. The production of the new nucleotide strands is catalyzed by DNA polymerase, an enzyme that adds new nucleotides to the 3' end of the growing strands. Because of the antiparallel orientation of the two DNA strands, the strands are formed differently. One strand, called the leading strand, is formed as a continuous strand, whereas the other strand, called the lagging strand, is formed in short segments called Okazaki fragments. The Okazaki fragments are then spliced by DNA ligase. As a result of DNA replication, two identical DNA molecules are produced. Each of the new DNA molecules has one strand of nucleotides derived from the original DNA molecule and one newly synthesized strand. Cell division. New cells necessary for growth and tissue repair are produced by cell division. A parent cell divides to form two daughter cells, each of which has the same amount and type of DNA as the parent cell. Because DNA determines cell structure and function, the daughter cells tend to have the same structure and perform the same function as the parent cell. During development and cell differentiation, however, the functions of daughter cells may differ from each other and from that of the parent cell. Cell division involves two major events: the division of the nucleus to form two new nuclei, and the division of the cytoplasm to form two new cells. Each of the new cells contains one of the newly formed nuclei. The division of the nucleus occurs by mitosis, and the division of the cytoplasm is called cytokinesis. There's a note for one of the illustrations, or there's 3 notes, so never mind, I'm going to skip those because I can't show the illustration. Mitosis. Mitosis is the division of the nucleus into two nuclei, each of which has the same amount and type of DNA as the original nucleus. The DNA which was replicated during interphase is dispersed as chromatin. During mitosis, the chromatin becomes very densely coiled to form compact chromosomes. These chromosomes are discrete bodies easily seen with a light microscope after staining the nuclei of dividing cells. Because the DNA has been replicated, each chromosome consists of 2 chromatids, which are attached at a single point called the centromere. Each chromatid contains a DNA molecule. As 2. End of page 92, beginning page 93. Daughter cells are formed the chromatids separate, and each is now called a chromosome. Each daughter cell receives one of the chromosomes. Thus, the daughter cells receive the same complement of chromosomes and DNA and are genetically identical. For convenience of discussion, mitosis is divided into 4 phases: prophase, metaphase, anaphase, and telophase. Although each phase represents major events, mitosis is a continuous process, and no discrete jumps occur from one phase to another. Learning the characteristics associated with each phase is helpful, but a more important concept is that each daughter cell obtains the same number and type of chromosomes as the parent cell. The major events of mitosis are summarized in figure 3.47, which I will not be showing. Cytokinesis. Cytokinesis is the division of the cytoplasm of the cell to produce 2 new cells. Cytokinesis begins in anaphase, continues through telophase, and ends in the following interphase. The first sign of cytokinesis is the formation of a cleavage furrow, or an indentation of the plasma membrane, which forms midway between the centrioles. A contractile ring composed primarily of actin filaments pulls the plasma membrane inward, dividing the cell into halves. Cytokinesis is complete when the membranes of the halves separate at the cleavage furrow to form 2 separate cells. Genetics. Genetics is the study of heredity, that is, those characteristics inherited by children from their parents. Although the environment can produce gene expression, people's physical characteristics and abilities are largely determined by their genetic makeup. Many of a person's abilities, susceptibility to disease, and even lifespan are influenced by the genes they inherit from their parents. Because many of the diseases caused by microorganisms now are preventable or treatable, diseases that have genetic basis are receiving more attention. An understanding of genetics is an important tool for medical professionals. Patients are asked questions about their family medical history to help diagnose many diseases. The family medical history also allows doctors to determine the probability that patients will develop certain diseases, such as heart disease, and at page 93, briefly skipping page 94 due to the illustrations and notes, resuming on page 95, cancer, and to suggest preventive measures. Recent advances in the field of genetics have shown how genes influence health and provided new methods for treating certain diseases. Some genetic diseases, such as hemophilia, have a simple pattern of inheritance, usually involving one or a few genes, and it is easy to predict whether a person will develop the disease. However, other genetic diseases have a more complex pattern of inheritance, involving many more genes. Mendelian genetics, the study of how certain genetic traits are passed from parent to offspring, has been used to determine an individual's risk of developing certain genetic diseases. Over the past 10 years, the human genome all of the genes in the human chromosomes have been sequenced. The results of this project have provided a wealth of information useful to the medical field. This information will allow researchers to better identify genes associated with particular diseases, to understand the biochemical relationship between genes and diseases, and to create new treatments for disease. The new genetic approach to the diagnosis and management of diseases is known as genomic medicine. I'm going to pause the reading there and return to page 94 to read the notes since, anyways. 1. Interface. This is talking about the cell division. There's 4 phases mentioned on page 93, but page 94 lists 5 and then 6 being when it's complete. So starting with number 1, interphase. Interphase is the time between cell divisions. DNA is found as thin threads of chromatin in the nucleus. DNA replication occurs during the S phase of interphase. Organelles other than the nucleus and centrioles duplicate during interphase. 2. In prophase, the chromatid condenses into chromosomes. Each chromosome consists of 2 chromatids joined at the centromere. The centrioles move to the opposite ends of the cell, and the nucleus and the nuclear envelope disappear. Microtubules form near the centrioles and project in all directions. Some of the microtubules end blindly and are called astral fibers. Others, known as spindle fibers, project toward an invisible line called the equator and overlap with fibers from opposite centrioles. 3. In metaphase, the chromosomes align in the center of the cell in association with the spindle fibers. Some spindle fibers are attached to kinetochores in the centromere of each chromosome. 4. In anaphase, the chromatids separate, and each chromatid is then referred to as a chromosome. Thus, when the centromeres divide, the chromosome number is doubled, and there are 2 identical sets of chromosomes. The chromosomes, assisted by the spindle fibers, move toward the centrioles at each end of the cell. Separation of the chromatids signals the beginning of anaphase, and by the time anaphase has ended, the chromosomes have reached the poles of the cell. Cytokinesis begins during anaphase as a cleavage furrow forms around the cell. 5. In telophase, migration of each set of chromosomes is complete. The chromosomes unravel to become less distinct chromatin threads. The nuclear envelope forms from the endoplasmic reticulum. The nucleoli form and cytokinesis continues to form 2 cells. 6. Mitosis is complete, and a new interphase begins. The chromosomes have unraveled to become chromatin. Cell division has produced 2 daughter cells, each with DNA that is identical to the DNA of the parent cell. Resuming on page 95. Mendelian genetics. Gregor Mendel, from 1822 to 1884, was an Austrian monk who made revolutionary discoveries about inheritance patterns in pea plants. For his discoveries, Mendel is known as the, quote, father of genetics, end quote. Mendel found that the transmission of traits such as purple versus white flowers from one generation of pea plants to another can be explained by discrete units he called, quote, heritable factors, end quote. He concluded that each pea plant has two heritable factors for a characteristic, such as a flower color. He proposed that during the production of gametes, or sex cells, each gamete receives one of these factors. Then two gametes combine to produce the next generation, so that each member of the next generation of pea plants has two heritable factors. Mendel's, quote, heritable factors, end quote, are now called genes. The genes an organism has for a given trait is called the genotype. The expression of the genes as traits is called the phenotype. Environmental effects can interact with gene expression to determine the phenotype of many traits. Even genetically identical twins can have different phenotypes because of environmental effects. For example, in addition to genes, height is affected by nutrition, and skin color by exposure to the sun. Knowledge of environmental effects can be used to improve our genetic potential and to prevent harmful effects. For example, a healthy diet can promote growth or help prevent diabetes, and not smoking can reduce the risk of developing cancer. Mendel proposed that genes can occur in dominant and recessive forms. Alternate forms of genes are now called alleles. By definition, the effects of a dominant allele for a trait mask the effects of the recessive allele for that trait. For example, in pea plants, the allele for purple flowers is dominant over the allele for white flowers. By convention, dominant alleles are indicated by uppercase letters, and recessive alleles are indicated by lowercase letters. For example, the letter P designates the dominant allele for purple flower color, and the letter P represents the recessive allele for white flower color. A plant with the capital P, lower p genotype has purple flowers because purple is dominant over white. An organism is homozygous for a trait if the two alleles for the trait are identical, whereas an organism is heterozygous for a trait if the two alleles for the trait are different. The possible genotypes and phenotypes for purple flower color are allele, capital P, genotype, homozygous dominant, phenotype purple, capital P, allele 2, capital P, lowercase p, genotype heterozygous, phenotype purple, allele 3, lowercase p, genotype homozygous recessive, phenotype white. Note that the recessive trait is expressed when it is not masked by the dominant trait. The relationship between genotype and phenotype can be understood on the molecular level. For example, There is a recessive trait in humans called albinism, which results in a lack of normal coloring of the skin, hair, and eyes. Several human genes produce the enzymes that are necessary for the synthesis of melanin, the pigments responsible for skin, hair, and eye color. See Chapter 5. An albino, a person with albinism, has light blonde or white hair and light-colored eyes and skin with shades of pink, blue, and yellow. The pink and blue colors result from blood seen in the eyes or through the skin. The yellow is from the natural accumulation of ingested yellow plant pigments in the skin. See Skin Color, Chapter 5. The normal alleles for the melanin synthesizing enzymes produce normal functional enzymes capable of catalyzing the steps in melanin synthesis. When normal enzymes are produced, a person has normal pigmentation. An abnormal allele for any one of the enzymes in the melanin pathway will produce a defective enzyme. The defective enzyme will block the pathway and stop the production of melanin. Type 1 albinism results from a defective enzyme that fails to initiate the synthesis of melanin from tyrosine, the normal allele of the gene for this enzyme. End of page 95, beginning page 96. Designated A, or capital A, is dominant over the abnormal allele, designated lowercase a. The possible genotypes and phenotypes for albinism are allele 1, capital A, genotype homozygous dominant, phenotype normal pigmentation. Allele 2, capital A, lowercase a, genotype heterozygous, phenotype normal pigmentation. Allele 3, lowercase a, genotype homozygous recessive, phenotype albino. A person with the genotype capital A has the phenotype of normal pigmentation because the functional enzyme for melanin synthesis is produced by both alleles. A person with the genotype capital A, lowercase a, has the phenotype of normal pigmentation, even though the lowercase a allele produces an abnormal non-functional enzyme for melanin synthesis. The capital A allele produces sufficient amounts of the normal enzyme so that pigmentation is normal. A person with the genotype lowercase a has the phenotype of albinism because only the abnormal non-functional enzyme for melanin synthesis is produced. Not all dominant traits are the normal condition, and not all recessive traits are abnormal. In many cases, the dominant trait is abnormal. For example, a person with polydactyly has extra fingers or toes. One allele for polydactyly, which results in extra fingers or toes, is dominant over the recessive normal allele, which results in the normal number of fingers or toes. Modern concepts of genetics. Although the existence of chromosomes was known in Mendel's time, the relationship between genes and chromosomes was not confirmed until T.H. Morgan was able to induce mutations, gene changes, in fruit flies in 1910. DNA was first isolated as the genetic material in 1944, and James Watson and Francis Crick resolved the structure of DNA in 1953, which led to an understanding of the genetic code and protein synthesis. The sequence of nucleotides of a human gene, specifically the gene that causes cystic fibrosis, was determined for the first time in 1989. A sequencing of 99% of the human genome was accomplished in 2003 with the completion of the Human Genome Project. See page 102. Chromosomes. In modern terminology, Mendel hypothesized that organisms have genes that control the expression of traits. Most genes, which are segments of DNA, occur in pairs of alleles. Chromosomes are made up of DNA and associated proteins found in the nuclei of somatic cells and gametes. Somatic cells are all the cells of the body except for the gametes. Examples of somatic cells are epithelial cells, muscle cells, neurons, fibroblasts, lymphocytes, and macrophages. In males, the gametes are sperm cells. In females, the gametes are oocytes, egg cells. See chapter 28. The normal number of chromosomes in a somatic cell is called the diploid, twofold number. The normal number of chromosomes in a gamete is the haploid, single number. In humans, the diploid number of chromosomes is 46. Chromosomes in diploid cells are paired, so humans have 23 pairs of chromosomes. The haploid number of chromosomes is 23. The 23 pairs of chromosomes are divided into autosomal and sex chromosomes. Humans have 22 pairs of autosomal chromosomes, which are all the chromosomes, but the sex chromosomes, and one pair of sex chromosomes, which determines the sex of the individual. Sex chromosomes are denoted as X or Y chromosomes. A normal female has 2 X chromosomes, XX, in each somatic cell. One X chromosome of a female is derived from her mother. The other is derived from her father. A normal male has 1 X chromosome and 1 Y chromosome, XY, in each somatic cell. The X chromosome of a male is derived from his mother. The Y chromosome is derived from his father. Genetic traits can be classified by the type of chromosome their alleles are located on and by whether their alleles are dominant or recessive. Thus, traits can be autosomal dominant or recessive, or they can be sex-linked dominant or recessive. For example, albinism is an autosomal recessive trait. A karyotype is a display of the chromosomes of a somatic cell during metaphase of mitosis. It is produced, end of page 96, beginning page 97, by photographing the cell's stained chromosomes through a microscope and arranging the photographed chromosomes in pairs. For convenience, the autosomal chromosomes are numbered from largest to smallest, 1 through 22. The sex chromosomes are denoted with an X or a Y. Note that the chromosome in a karyotype replicated chromosome, that is, it has two chromatids. The chromatids of each chromosome are attached at the centromere so that each replicated chromosome appears as a single structure. Chromosome pairs are called homologous pairs, and each member of the pair is called a homolog. One homolog is derived from a person's father, and the other is derived from a person's mother. A genome consists of all the genes found in the haploid number of chromosomes from one parent. The combined genomes from both parents are responsible for all of the person's genetic traits. Each gene of the genome occupies a specific locus or location on a chromosome. The locus on one chromosome may have one allele type, whereas the locus on its homolog may have another allele type. Multiple alleles. Mendel believed that alleles came in two forms, dominant and recessive. It is now known that alleles can exist in many forms, called multiple alleles. An individual has only two alleles for a given gene, one on each homologous chromosome. At the population level, however, there can be many forms of an allele. Differences in alleles arise by mutation. See page 100, in which the DNA nucleotide sequence is altered. Many alleles are possible in a population because any change in the nucleotide sequence of DNA, even of one base pair, potentially produces a different allele. A different form of an allele is called an allelic variant, a mutated allele, gene, or a polymorphism, many forms. Allelic variants can result in either no effect on the phenotype or minor to major phenotypic changes. Allelic variants at a locus may encode for different sequences of amino acids and proteins. Recall from Chapter 2 that the sequence of amino acids in a protein affects the shape of the protein, including the protein's domain, which is the functional part of the protein. A mutated allele that causes an amino acid change in a protein that does not significantly change protein shape may not affect the phenotype. The greater the effect of an amino acid change on a protein's shape, however, the greater the effect on the protein's function, and the greater the effect on the phenotype. Millions of allelic variants have been identified. The significance of most of them is unknown. The allelic variant present at a locus can affect a person's phenotype. For example, phenylketonuria, PKU, phenylketonurica, is an autosomal recessive trait with multiple alleles. On chromosome 12 is a gene that encodes for an enzyme that converts the amino acid phenylalanine to the amino acid tyrosine. If phenylalanine is not converted to tyrosine, high levels of phenylalanine can accumulate, leading to brain cell damage. The more defective the enzyme, the greater the accumulation of phenylalanine, and the greater the damage to the brain in most cases. over 400 disease-causing allelic variants of the end of page 97, beginning page 98, PKU gene are known. The severity of PKU depends in part on which two allelic variants are present. The less severe of the two alleles determines the severity of the disorder. Symptoms of untreated PKU can vary from profound mental retardation to nearly normal mental abilities. Thus, even though PKU is an autosomal recessive trait, the expression of the recessive condition exhibits considerable variability because of multiple alleles. Furthermore, reducing phenylalanine in the diet can prevent the development of mental retardation. Gene dominance. In Mendel's experiments with pea plants, each dominant allele produced enough of its protein product to cause the maximum phenotypic response in the heterozygote. Thus, the homozygous dominant and heterozygote had the same phenotype, that is, purple flowers. This is called complete dominance. It is now known that many other types of gene expression exist. Complete dominance is at one end of a continuum of genetic expression. At the other end is codominance, and between them is incomplete dominance. In codominance, two alleles at the same locus are expressed so that separate distinguishable phenotypes occur at the same time. ABO blood types are an example of codominance. A gene with 3 alleles on chromosome 9 encodes for enzymes that add sugar molecules to certain carbohydrates found on the surface of red blood cells. The carbohydrates are part of glycoproteins called A and B. Hydrates are called A and B antigens. See chapter 19. These carbohydrates consist of different sugars joined together. Traditionally, in designating alleles for blood type, the A and B alleles are superscripted to a capital letter, quote, I, end quote, and the O allele is designated with the lowercase letter, quote, I, end quote. The IA allele encodes for an enzyme that adds a particular sugar to the ends of the carbohydrate, producing the A antigen. The IB allele encodes for a different enzyme that adds a different sugar to the end of the carbohydrate, producing the B antigen. The lowercase I allele encodes for no functional enzyme and therefore is recessive to A and B. The possible genotypes and phenotypes for the ABO blood group are genotype, capital I, capital A, capital I, capital A, or capital I, capital A, lowercase I. Phenotype, type A blood, A antigen only. Genotype, capital I, capital B, capital I, capital B, or capital I, capital B, lowercase I. Phenotype, type B blood, B antigen only. Genotype, capital I, capital A, capital I, capital B. Phenotype, type AB blood, A and B antigens. Genotype, lowercase I, lowercase I. Phenotype, type O blood, neither A nor B antigen. Type A blood results from the expression of only the capital I, capital A allele. Type B blood from the expression of only the capital I, capital B allele. And type O blood from the expression of neither the capital I, capital A allele, nor the capital I, capital B allele. Type AB blood illustrates the codominance and results from the expression of both the capital I, capital A allele and the capital I, capital B allele at the same time. In incomplete dominance, the dominant allele does not completely mask the effects of the recessive allele in the heterozygote. The heterozygote produces less of the protein product than the homozygous dominant and has phenotypic characteristics intermediate between the homozygous dominant and the homozygous recessive. For example, beta thalassemia is a disorder of a gene on a chromosome 11. It affects the synthesis of beta-globulin polypeptide chains, which are part of the hemoglobin in red blood cells. Hemoglobin is a protein that transports oxygen. If normal amounts of the beta-globulin polypeptide are produced, the beta-globulin peptides join with other proteins to form hemoglobin. If lower than normal amounts of beta-globulin polypeptide are synthesized, lower than normal amounts of hemoglobin are produced. In the homozygous dominant state, two normal alleles for beta-globulin synthesis are present. Normal amounts of hemoglobin are produced, and the phenotype is normal, meaning that there is normal transport of oxygen. In the homozygous recessive condition, called major thalassemia, two abnormal alleles are present, and much lower than normal amounts of beta-globulin polypeptide chains are synthesized. The result is severe anemia, which is a deficiency of hemoglobin in the blood. Symptoms include pallor, weakness, fatigue, and spleen enlargement. Blood transfusions are necessary to maintain hemoglobin levels. In the heterozygous condition called minor thalassemia, one normal allele and one abnormal allele are present. The reduction of hemoglobin is intermediate between the normal phenotype and major thalassemia, and mild anemia results. Polygenic traits. In some cases, one gene, usually with two alleles, determines one phenotype. In other cases, many genes determine a phenotype. These polygenic traits result from the interactions of many genes. Although the genes contribute to the phenotype without being dominant or recessive to each other, each gene has its own characteristics, such as multiple alleles or incomplete dominance. Some of the genes may be more important than others for the expression of the phenotype, and the genes can be on different chromosomes. The result of all the gene interactions is a phenotype with a great amount of variability. Examples of polygenic traits are height, intelligence, eye color, and skin color. Even though a complex combination of genes determines a polygenic trait, a defect in one of them can sometimes have dramatic effect on phenotype. For example, even though many genes contribute to skin color, one defective gene can eliminate skin color completely, resulting in albinism. Sex-linked traits. Sex-linked traits are traits affected by genes on the sex chromosomes. X-linked traits are on the X chromosome, and Y-linked traits are on the Y chromosome. Most sex-linked traits are X-linked, whereas only a few Y-linked traits exist, mainly because the Y chromosome is very small. An example of an X-linked recessive trait is hemophilia A, classic hemophilia, in which the ability to produce certain blood clotting factors is not present. See Chapter 19. Consequently, clotting is impaired and persistent bleeding occurs either spontaneously or as a result of an injury. Traditionally, X-linked genes are designated by letters superscripted to the letter quote X end quote. The possible genotypes and phenotypes for hemophilia A are genotype capital X, capital H, capital X, capital H, or capital X, capital H, capital X, lowercase h. Phenotype is normal female. Next, genotype capital H, lowercase h, capital H, lowercase h. Phenotype hemophilic female. Next, genotype capital X, capital H, capital Y. Phenotype normal male. Next, genotype capital X, lowercase h, capital Y. Phenotype hemophilic male. Note, and that is the page of N98, the end of page 98, now the beginning of page 99. Note that a female must have both recessive alleles, capital H, lowercase h, capital H, lowercase h, to exhibit hemophilia, whereas a male, because he has only one X chromosome, has hemophilia if he has only one of the recessive alleles. For this reason, X-linked recessive traits are seen more frequently in males than females. Meiosis and the transmission of genes. Gametes are haploid cells that are derived from diploid cells. In meiosis, gametes are produced that have one homolog from each of the homologous pairs of chromosomes. Therefore, gametes have 1/2 the number of chromosomes and 1/2 the alleles as the original diploid cells. Before meiosis begins, DNA replication occurs so that replicated chromosomes are formed, each consisting of two chromatids. As a result of the first meiotic division, each daughter cell receives one replicated member of a homologous pair. No replication of DNA takes place between the first and second meiotic divisions. During the second meiotic division, the chromatids of each chromosome separate. As a result of this second meiotic division, each cell receives one chromatid from each chromosome. That single chromatid is now referred to as a chromosome. Each gamete now has a haploid number of chromosomes. See Chapter 28 for a more detailed discussion of gamete production. In Figure 3.52, which I won't show, the alleles for albinism are shown on the homologous chromosomes. On one homolog, the normal dominant allele is indicated with an uppercase A. On the other homolog, the abnormal allele is indicated with a lowercase a. End of page 99, beginning page 100. The probability of the transmission of alleles to the next generation can be determined using a Punnett square if the genotypes of the parent are known. The possible alleles found in the gametes of one parent form the rows of the square, and the possible alleles found in the gametes of the other parent form the columns. The body of the Punnett square shows all possible combinations of the parent's gametes. For example, suppose both parents are heterozygous, capital A, lowercase a, for albinism. The heterozygous parents can produce two types of gametes. One gamete has the capital A allele, and the other has the lowercase a allele. The probability that the child will be homozygous dominant, capital A, is 1 out of 4. Heterozygous, capital A, lowercase a, 2 out of 4. And homozygous recessive, capital A, lowercase a, is 1 out of 4. Albinism is an autosomal recessive trait. The homozygous dominant and heterozygous conditions result in normal pigmentation, whereas the homozygous recessive condition results in albinism. Both heterozygous parents have normal pigmentation, but there is a 1 out of 4 chance that any one of their children will be an albino. A carrier for a recessive trait is a person who is heterozygous for that trait, with 1 normal allele and 1 disorder-causing allele. The carrier does not exhibit the disorder, but can pass it on to his or her children. Genetic disorders. A genetic disorder is a failure of structure, function, or both as a result of abnormalities in a person's genetic makeup, that is, his or her DNA. Humans suffer from a variety of genetic disorders, some of which are highlighted throughout the remainder of this book. Genetic disorders determined by one gene are described by Mendelian characteristics, for example, as autosomal recessive. In some cases, this type of description is accurate, but in other cases, the reality is more complex. For example, PKU is said to be autosomal recessive, but PKU has multiple alleles which result in a spectrum of defects. As you study the genetic disorders in later chapters, keep in mind the concepts discussed in this chapter. Genetic disorders involve either a single gene or an entire chromosome, as in the case of aneuploid, euploidy. Table 3.4. Recall that a mutation is a change in a gene that usually involves a change in the number or kinds of nucleotides composing the DNA. See Chapter 2. Mutations are known to occur by chance, randomly without known cause, but they can also be caused by chemicals, radiation, and viruses. Agents that cause mutations are called mutagens. In most cases, a specific cause of the mutation cannot be determined. Once a mutation has occurred, however, the abnormal trait can be passed from one generation to the next. A mutation involving a single nucleotide change is called a point mutation. Mutations that change the sequence or number of nucleotides involve changes in chromosome structure, and therefore are called structural mutations. Structural mutations can involve deletions, the loss of part of a chromosome. Duplications involve, duplications, the addition of a copied section of a chromosome. Translocations, which part of one chromosome becomes attached to another chromosome, and inversions, in which the DNA sequence of a section of a chromosome is reversed. Furthermore, segregation errors that occur during meiosis result in changes in the chromosome number in the gametes. As the chromosomes separate during meiosis, two members of a homologous pair may become, quote, sticky, end quote, and not segregate as they normally do. As a result, one of the daughter cells receives both chromosomes, and the other daughter cell receives none. This event is called non-disjunction. When the gametes are fertilized, the resulting zygote has either 47 chromosomes or 45 chromosomes, rather than the normal 46, a condition called aneuploidy. Aneuploidies include monosomies, in which a chromosome is missing, or trisomies, in which an extra chromosome is present. Aneuploidies are usually lethal and is one reason for a high rate of early embryo loss. Down syndrome, or trisomy 21, is a type of aneuploidy in which 3 chromosomes, 21, are present. A syndrome is a set of signs and symptoms occurring together as the result of a single cause, such as a single mutation or one extra chromosome, a trisomy. Down syndrome is an example of an aneuploidy that is not always lethal. However, individuals with this genetic disorder exhibit physical and mental developmental problems, as well as an increased probability of developing certain cancers. These individuals also have shortened lifespans. End of page 100. Beginning page 101. Table 3.4. Genetic disorders. Disorder and description. Disorder. Dominant traits. Achondroplasia. Description. Dwarfism characterized by shortening of the upper and lower limbs. Disorder. Huntington chorea. Description. severe degeneration of the basal nuclei and frontal cerebral cortex, characterized by purposeless movements and mental deterioration. Onset is usually between 40 and 50 years of age. Disorder: hypercholesterolemia. Description: elevated blood cholesterol levels that contribute to atherosclerosis and cardiovascular disease. Disorder: Marfan syndrome. Description: abnormal connective tissue, resulting in increased height, elongated digits, and weakness in the aortic wall. Disorder: neurofibromatosis. Description: small pigmented lesions, café au lait spots in the skin, and disfiguring tumors, non-cancerous, caused by the proliferation of Schwann cells along nerves. Disorder: osteogenesis imperfecta. Description: abnormal collagen synthesis resulting in brittle bones that break repeatedly. Recessive traits. Disorder: albinism. Description: lack of an enzyme necessary to produce the pigment melanin, characterized by lack of skin, hair, and eye coloration. Disorder: cystic fibrosis. Description: impaired transport of chloride ions across plasma membranes, resulting in excessive production of thick mucus, which blocks the respiratory and gastrointestinal tract. The most common fatal genetic disorder. Disorder: phenylketonuria. Description: lack of an enzyme necessary to convert the amino acid phenylalanine to the amino acid triosine. An accumulation of phenylalanine leads to mental retardation. Disorder: severe combined immune deficiency. Description: inability to form white blood cells, B cells, T cells, and phagocytes, necessary for an immune system response. Disorder: sickle cell disease. Description: inability to produce normal hemoglobin, resulting in abnormally shaped red blood cells that clog capillaries or rupture. Disorder: Tay-Sachs disease. Description: lack of the enzyme necessary to break down certain fatty substances. An accumulation of fatty substances impairs action potential propagation, resulting in deterioration of mental and physical functions, and death by 3 to 4 years of age. Disorder: thalassemia. Description: decreased rate of hemoglobin synthesis, resulting in anemia, enlargement of the spleen, increased number of cells in red bone marrow, and congestive heart failure. Sex-linked traits. Disorder: Duchenne muscular dystrophy. Description: deletion or alteration of part of the X chromosome resulting in progressive weakness and wasting of muscles. Disorder: hemophilia. Description: most commonly failure to produce blood clotting factors caused by a recessive gene, resulting in prolonged bleeding. Disorder, red-green color blindness. Description, most commonly deficiently in functional green sensitive cones, caused by a recessive gene, inability to distinguish between red and green colors. Chromosomal diseases. Down syndrome, caused by having 3 chromosomes 21, resulting in mental retardation, short stature, and poor muscle tone. Klinefelter syndrome, caused by 2 or more X chromosomes in a male, XXY, resulting in small testes, sterility, and development of female-like breasts. Last disorder, Turner syndrome. Description, caused by having 1 X chromosome, resulting in immature uterus, lack of ovaries, and short stature. That's the end of table 3.4. Next, sex chromosome abnormalities. A wide range of sex chromosome abnormalities exist. The presence of a Y chromosome makes a person genetically a male, and the absence of a Y chromosome makes a person genetically a female, regardless of the number of X chromosomes. Individuals with X0, Turner syndrome, XX, XXX, or XXXX karyotypes are therefore females, and individuals with XY, XXY, XXXY, or XYY karyotypes are males. A Y0 condition is lethal because the genes on the X chromosome are necessary for survival. Secondary sexual characteristics are usually underdeveloped in both the X0 female and the XXY male, called Klinefelter syndrome, and additional X chromosomes, XXXX or XXXY, are often associated with some degree of mental retardation. As a result of hormonal imbalances, the morphological features, phenotype, may be reversed from the genetic constitution, genotype. Resuming the main reading. Cancer usually results from a series of somatic mutations that cause uncontrolled cell division and changes in the normal function of cells. The genetics of cancer is discussed in the quote, clinical genetics, end quote, essay in chapter 4. End of page 101, beginning page 102. Clinical Genetics, the Human Genome Project. The human genome consists of all the genes found in one homologous set of human chromosomes. It is estimated that humans have 25,000 to 30,000 genes. A genomic map is a description of the DNA nucleotide sequences of the genes and their locations on the chromosomes. Celera is a private corporation working on the Human Genome Project, announced on February 12, 2001, that it had completed sequencing 99% of the genome. Online Mendelian Inheritance in Man, OMIM, is a database that contains information about many human genes and genetic disorders. www.ncbi.nlm.gov/OMIM. Armed with a knowledge of the human genome and its effects on a person's physical, mental, and behavioral abilities, medicine and society will be transformed in many ways. Medicine, for example, will shift emphasis from the curative to the preventive. The disorder or diseases a person is likely to develop can be prevented or their severity lessened. When prevention is not possible, knowledge of the enzymes or other molecules involved in a disorder may result in new drugs and techniques that can compensate for the genetic disorder. Knowledge of the genes involved in a disorder may result in gene therapy or genetic engineering that repairs or replaces defective genes, resulting in cures of or treatments for genetic disorders. Despite the great promise of benefits from the Human Genome Project, the knowledge produced has raised a number of ethical and legal questions for society. Should a person's genomic information be public knowledge? Should persons with a genome that predisposes them to cancer or behavioral disorders be barred from certain types of employment or be refused medical insurance because they are at high risk? Can a person demand to know a prospect mate's genome? Should parents know the genome of their fetus and be allowed to make decisions regarding abortion based on this knowledge? Should the same genetic engineering techniques that provide alteration of the genome to cure genetic disorders be used to create genomes that are deemed to be superior? Such questions raise the specter of genetic discrimination. So that is the end of chapter 3, and part of the section on clinical genetics, describing the Human Genome Project, goes from page 102 and it ends on page 103. But the majority of page 102 has a diagram of chromosome pairs and different notes on the bottom. And then the majority of page 103, about 2/3 of the page, covers the summary, which I will actually go ahead and start reading now. Summary. 1. The plasma membrane forms the outer boundary of the cell. 2. The nucleus directs the activities of the cell. 3. Cytoplasm between the nucleus and plasma membrane is where most cell activities take place. Functions of the cell, page 56. 1. Cells metabolize and release energy. 2. Cells synthesize molecules. 3. Cells provide a means of communication. 4. Cells reproduce and provide for inheritance. How we see cells, page 58. 1. Light microscopes allow us to visualize the general features of cells. 2. Electron microscopes allow us to visualize the fine structure of cells. Plasma membrane, page 58. 1. The plasma membrane passively or actively regulates what enters or leaves the cell. 2. The plasma membrane is composed of a phospholipid bilayer in which proteins are suspended. Fluid mosaic model. Membrane lipids, page 58. Lipids give the plasma membrane most of its structure and some of its function. Membrane proteins, page 58. 1. Membrane proteins function as marker molecules, attachment proteins, transport proteins, receptor proteins, and enzymes. 2. Transport proteins include channel proteins, carrier proteins, transporters, and ATP-powered pumps. 3. Some receptor molecules are linked to and control channel proteins. 4. Some receptor molecules are linked to G protein complexes, which in turn control numerous cellular activities. Movement through the plasma membrane, page 64. 1. Lipid soluble molecules pass through the plasma membrane readily by dissolving in the lipid bilayer. 2. Small molecules pass through the membrane through membrane channels. Most channels are positively charged, allowing negatively charged ions and neutral molecules to pass through more readily than positively charged ions. 3. Large polar substances, example, glucose and amino acids, are transported through the membrane by carrier molecules. 4. Larger pieces of material enter cells in vesicles. Diffusion. 1. Diffusion is the movement of a substance from an area of higher solute concentration to one of lower solute concentration, down a central concentration gradient. 2. The concentration gradient is the difference in solute concentration between 2 points divided by the distance separating the points. 3. The rate of diffusion increases with an increase in the concentration gradient, an increase in temperature, a decrease in molecular size, and a decrease in viscosity. 4. The end result of diffusion is a uniform distribution of molecules. 5. Diffusion requires no expenditure of energy. Osmosis. 1. Osmosis is the diffusion of water, solvent, across a selectively permeable membrane. 2. Osmotic pressure is the force required to prevent the movement of water across a selectively permeable membrane. End of page 103. Beginning page 104. 3. Isomotic solutions have the same concentration of solute particles. Hypersomatic solutions have a greater concentration, and hypersomatic solutions have a lesser concentration of solute particles than a reference solution. 4. Cells placed in an isotonic solution neither swell nor shrink. In a hypertonic solution, they shrink, crenate. In a hypertonic solution, they swell and may burst. Lyse. Filtration. 1. Filtration is the movement of a liquid through a partition with holes that allows the liquid, but not everything in the liquid, to pass through them. 2. Liquid movement results from a pressure difference across the partition. Mediated transport. 1. Mediated transport is the movement of a substance across a membrane by means of a transport protein. The substances transported tend to be large, water-soluble molecules. The transport protein has binding sites that bind with either a single molecule or a group of similar molecules. This selectiveness is called specificity. Similar molecules can compete for transport proteins, with each reducing the rate of transport of the other. Once all the transport proteins are in use, the rate of transport cannot increase further. Saturation. 2. 3 kinds of mediated transport can be identified. Facilitated diffusion moves substances down their concentration gradient and does not require energy, ATP, expenditure. Active transport can move substances against their concentration gradient and requires ATP. An exchange pump is an active transport mechanism that simultaneously moves 2 substances in opposite directions across the plasma membrane. In secondary active transport, an ion is moved across the plasma membrane by active transport, and the energy produced by the ion diffusing back down its concentration gradient can transport another molecule, such as glucose, against its concentration gradient. Endocytosis and exocytosis, page 71. 1. Endocytosis is the bulk movement of materials into cells. Phagocytosis is the bulk movement of solid material into cells by the formation of a vesicle. Pinocytosis is similar to phagocytosis, except that the ingested material is much smaller or is in solution. 2. Exocytosis is the secretion of materials from cells by vesicle formation. 3. Endocytosis and exocytosis use vesicles, can be specific, receptor-mediated endocytosis for the substance transported and require energy. Cytoplasm, page 76. The cytoplasm is the material outside the nucleus and inside the plasma membrane. Cytosol. 1. Cytosol consists of a fluid part, the site of chemical reactions, the cytoskeleton and cytoplasmic inclusions. 2. The cytoskeleton supports the cell and enables cell movements. It consists of protein fibers. Microtubules are hollow tubes composed of the protein tubulin. They form spindle fibers and are components of centrioles, cilia, and flagella. Actin filaments are small protein fibrils, that provide structure to the cytoplasm or cause cell movements. Intermediate filaments are protein fibers that provide structural strength to cells. 3. Cytoplasmic inclusions, such as lipochromes, are not surrounded by membranes. The nucleus and cytoplasmic organelles, page 77. Organelles are subcellular structures specialized for specific functions. Nucleus. 1. The nuclear envelope consists of a double membrane with nuclear pores. 2. DNA and associated proteins are found inside the nucleus as chromatin. 3. DNA is the hereditary material of the cell. It controls the activities of the cell by producing proteins through RNA. 4. A gene is a portion of a DNA molecule. Genes determine the proteins in a cell. 5. Nucleoli consist of RNA and proteins and are the sites of ribosomal subunit assembly. Ribosomes. 1. Ribosomes consist of small and large subunits manufactured in the nucleolus and assembled in the cytoplasm. 2. Ribosomes are the site of protein synthesis. 3. Ribosomes can be free or associated with the endoplasmic reticulum. Endoplasmic reticulum. 1. The endoplasmic reticulum is an extension of the outer membrane of the nuclear envelope. It forms tubules or sacs, cisternae, throughout the cell. 2. The rough endoplasmic reticulum has ribosomes and a site of protein synthesis and modification. 3. The smooth endoplasmic reticulum lacks ribosomes and is involved in lipid production, detoxification, and calcium storage. Golgi apparatus. The Golgi apparatus is a series of closely packed, modified cisternae that function to modify, package, and distribute lipids and proteins produced by the endoplasmic reticulum. Secretory vesicles. Secretory vesicles are membrane-bound sacs surrounded by membranes that carry substances from the Golgi apparatus to the plasma membrane, where the contents of the vesicle are released by exocytosis. Lysosomes. 1. Lysosomes are membrane-bound sacs containing hydrolytic enzymes. Within the cell, the enzymes break down phagocytized material and non-functional organelles. Autophagia. 2. Enzymes released from the cell by lysis, or enzymes secreted from the cell, can digest extracellular material. Peroxisomes. Peroxisomes are membrane-bound sacs containing enzymes that digest fatty acids and amino acids, as well as enzymes that catalyze the breakdown of hydrogen peroxide. End of page 104, beginning page 105. Proteasomes. Proteasomes are large multi-enzyme complexes, not bound by membranes, which digest selected proteins within the cell. Mitochondria. 1. Mitochondria are the major sites of the production of ATP, which is used as an energy source by cells. 2. The mitochondria have a smooth outer membrane and an inner membrane that is infolded to produce cristae. 3. Mitochondria contain their own DNA, can produce some of their own proteins, and can replicate independently of the cell. Centrioles and spindle fibers. 1. Centrioles are cylindrical organelles located in the centrosome, a specialized zone of the cytoplasm. The centrosome is the site of microtubule formation. 2. Spindle fibers are involved in the separation of chromosomes during cell division. Cilia and flagella. 1. Movement of materials over the surface of the cell is facilitated by cilia. 2. Flagella, much longer than cilia, propel sperm cells. Microvilli. Microvilli increase the surface area of the plasma membrane for absorption or secretion. Genes and gene expression, page 86. 1. During transcription, information stored in DNA is copied to form mRNA. 2. During translation, the mRNA goes to ribosomes, where it directs the synthesis of proteins. Transcription. 1. DNA unwinds and, through nucleotide pairing, produces pre-mRNA. Transcription. 2. Introns are removed and exons are spliced by spliceosomes during post-transcriptional processing. 3. Modifications to the ends of mRNA also occur during post-transcriptional processing. Genetic code. The genetic code, which specifies amino acids, consists of codons, which are sequences of 3 nucleotides in mRNA. Translation. 1. The mRNA moves through the nuclear pores to ribosomes. 2. Transfer RNA, tRNA, which carries amino acids, interacts at the ribosome with mRNA. The anticodons of tRNA bind to the codons of mRNA, and the amino acids are joined to form a protein. Translation. 3. Proproteins, some of which are proenzymes, are modified into proteins, some of which are enzymes, during post-translational processing. Regulation of genetic expression. 1. Cells become specialized because of inactivation of certain parts of the DNA molecule and activation of other parts. 2. The level of DNA activity, and thus protein production, can be controlled internally or can be affected by regulatory substances secreted by other cells. Cell life cycle, page 91. The cell life cycle has 2 stages: interphase and cell division. Interphase. Interphase is the period between cell divisions. This is the time of DNA replication. DNA replication. DNA unwinds and each strand produces a new DNA molecule during replication. Cell division. 1. Cell division includes nuclear division and cytoplasmic division. 2. Mitosis is the replication of the nucleus of the cell, and cytokinesis is division of the cytoplasm of the cell. 3. Mitosis is a continuous process divided into 4 phases. Prophase. Chromatin condenses to become visible as chromosomes. Each chromosome consists of 2 chromatids joined at the centromere. Centrioles were moved to opposite poles of the cell, and astral fibers and spindle fibers form. Nucleoli disappear, and the nuclear envelope degenerates. Metaphase. Chromosomes align at the equatorial plane. Anaphase. The chromatids of each chromosome separate at the centromere. Each chromatid then is called a chromosome. The chromosomes migrate to opposite poles. Telophase. Chromosomes unravel to become chromatin. The nuclear envelope and nucleoli reappear. 4. Cytokinesis begins with the formation of the cleavage furrow during anaphase. It is complete when the plasma membrane comes together at the equator, producing 2 new daughter cells. Genetics, page 93. 1. Genetics is the study of heredity, that is, characteristics inherited by children from their parents. 2. Genomic medicine uses an understanding of the biochemical relationship between genes and disease to diagnose and manage disease. Mendelian genetics. 1. The genes an organism has for a given trait is called the genotype. The expression of the genes is called the phenotype. 2. Alleles are alternate forms of genes. A dominant allele masks the effects of the recessive allele for the same trait. 3. An organism homozygous for a trait has 2 identical alleles for the trait, whereas an organism heterozygous for a trait has 2 different alleles for the trait. Modern concepts of genetics. 1. Chromosomes. Somatic cells have a diploid number of chromosomes, whereas gametes have a haploid number. In humans, the diploid number is 46, and the haploid number is 23. Humans have 22 pairs of autosomal chromosomes and 1 pair of sex chromosomes. Females have the sex chromosomes XX, and males have XY. A karyotype is a display of the chromosomes of a somatic cell during metaphase of mitosis. Chromosome pairs are called homologous chromosomes. 2. The genome consists of all the genes found in the haploid number of chromosomes from one parent. End of page 105, beginning page 106. 3. Each gene occupies a specific locus or location on a chromosome. 4. Alleles exist in many different forms, called multiple alleles. 5. Gene dominance. In complete dominance, the dominant allele masks the effects of the recessive allele. In codominance, two alleles at the same locus are expressed so that separate distinguishable phenotypes occur at the same time. In incomplete dominance, the dominant allele does not completely mask the effects of the recessive allele. 6. Polygenic traits result from the interaction of many genes. 7. Sex-linked traits. Sex-linked traits are traits affected by genes on the sex chromosomes. X-linked traits are affected by genes on the X chromosome, and Y-linked traits are traits affected by genes on the Y chromosome. X-linked traits are seen more frequently in males than in females because males have only one X chromosome. Meiosis and the transmission of genes. 1. Meiosis results in the production of gametes. 2. A Punnett square can determine the probability of the transmission of alleles to the next generation. 3. A carrier for a recessive trait is a person who is heterozygous for the trait, with one normal allele and one disorder-causing allele. Genetic disorders. A mutation is a change in the number or kinds of nucleotides in DNA. 2. Some genetic disorders result from an abnormal distribution of chromosomes during gamete formation. 3. Cancer results from a series of somatic mutations. And that is the end of the summary of Chapter 3. The rest of page 106, page 107, and page 108 are review and comprehension questions, which I'm just going to skip. And for the next video, I plan to start back on page 109 to begin Chapter 4, Histology, the study of tissues.